DNA molecule, we have seen, has two polynucleotide chains. Two polynucleotide chains, and these two polynucleotide chains, they are coiled around each other. Coiled around each other to form a double helix. The two polynucleotide chains of DNA run in opposite directions. ಏನು ಹಾಗಂದರೆ, ಸಪೋಸ್ this is one polynucleotide chain of DNA. This is another polynucleotide chain of DNA. Spirally coiled around each other. For this chain, this is the 3' end, and this is the 5' end, whereas for the other chain, this is the 3' end, this is the 5' end. In other words, it means the formation of this chain has proceeded in this direction. The first nucleotide is here and the last nucleotide is here, whereas the formation of this chain has proceeded in the opposite direction. So the first nucleotide will be here and the last nucleotide will be here. This 3' 5' this 3' 5' represents the linkage called the direction of linkage called phosphodiester linkage. Phosphodiester linkage between the two successive nucleotides. So that linkage is called phosphodiester linkage. The bond that is established is called phosphodiester bond. The direction of that linkage is 3' to 5'. The uprights of the helix, the upright portion of the helix is represented by phosphate and sugar alternately. So from here to here, throughout the length of the upright column, you have phosphate, sugar, phosphate, sugar, phosphate, sugar, and so on. Similarly, in the other chain, phosphate, sugar, phosphate, sugar, phosphate, sugar, and so on. Whereas the horizontal columns are represented by nitrogen bases. Nitrogen bases. So wherever there is a sugar, there is a nitrogen base. Wherever there is a sugar, there is a nitrogen base. So uprights of the helix formed by phosphate and sugar, horizontal columns formed by the nitrogen bases. Watson and Crick found that the two polynucleotide chains are separated by a distance of 20 angstrom units. 20 angstrom units throughout. So the average distance between the two chains throughout is 20 angstrom units. 1 angstrom unit is equal to 1 by 1000 of a millimeter. This is one important information which the double helix model could give us. Another important information is one full coil is called gyre. So from here to here it is one gyre, from here to here it is one gyre, and so on. So one full coil of the polynucleotide chain is called gyre. Watson and Crick found that each gyre measures 34 angstrom units in length. So So the length of a gyre is 34 angstrom units. Then another important observation is that the distance between any two successive sugar molecules is 3.4 angstrom units. It means the distance between successive sugar molecules, this sugar molecule and this sugar molecule, is 3.4 angstrom units. Similarly, between this sugar and this sugar, it is 3.4 angstrom units. Therefore, a very important piece of information that we can get here is that each gyre, each full coil, therefore accommodates 10 nucleotides. Each gyre accommodates 10 nucleotides. Length of the gyre is 34 angstrom units. Distance between any two successive sugar molecules is 3.4 angstrom units. So 34 divided by 3.4, 10. That means in this coil there will be 10 sugar molecules. Therefore, there can be 10 nucleotides. So each gyre accommodates 10 nucleotides. The most significant feature in the structure of the DNA molecule is the relationship between the nitrogen bases that occupy the same position in the two opposite chains. This is called specific base pairing. This idea of specific base pairing was first described by a biologist by name Chargaff in the year 1950, and this idea was incorporated in describing the structure of the DNA molecule by Watson and Crick. According to this specific base pairing, a purine pairs only with a pyrimidine. What does it mean? A purine pairs only with a pyrimidine means if this nitrogen base is a purine, this nitrogen base has to be a pyrimidine. If this is a pyrimidine, this should be a purine. So a purine pairs only with a pyrimidine. Similarly, a pyrimidine pairs only with a purine, vice versa. Among purines, we have seen there are two purines. Among purines, there are two purines, adenine and guanine. Adenine pairs only with thymine. Adenine pairs only with thymine. Adenine is a purine, thymine is a pyrimidine. So if this is adenine, this should be a thymine, or a thymine pairs only with adenine. In the same way, another purine, adenine is one purine, guanine is another purine. Guanine pairs only with cytosine, and similarly cytosine pairs only with guanine. This is what is called specific base pairing. Purine only with a pyrimidine, pyrimidine only with a purine. Among purines, adenine only with thymine, guanine only with cytosine. Between adenine and thymine, imagine there is adenine here, there is thymine here. Between adenine and thymine, there are two weak hydrogen bonds. Adenine and thymine are linked by two weak hydrogen bonds. Similarly, between guanine and cytosine, there are three weak hydrogen bonds. Symbolically, we can represent like this: phosphate, sugar, phosphate, sugar. This is adenine. Here phosphate, sugar, phosphate, sugar. Suppose here it is thymine. Between adenine and thymine and adenine, suppose you have thymine here, adenine here, again two weak hydrogen bonds. Between guanine and cytosine, or cytosine and guanine, three weak hydrogen bonds. Then, because of this highly specific base pairing, total amount of purines A plus G, adenine plus guanine, will be equal to total amount of A plus G will be equal to total amount of pyrimidines C plus T. Putting it the other way, A plus G will be equal to C plus T. and the ratio of AT, A plus T by C plus G. The ratio of A plus T by C plus G will be always a constant for a given species. So these are the salient features that the base pairing gives us. Purine pairs only with pyrimidine, adenine pairs only with thymine, guanine pairs only with cytosine. Between adenine and thymine there are 2 weak hydrogen bonds. Between guanine and cytosine there are 3 weak hydrogen bonds. Total amount of purines is equal to total amount of pyrimidines. A plus G is equal to C plus T, and the ratio of A plus T by C plus G will be a constant for a given species. So let us make this idea of base pairing little more clear by enlarging a small portion of the DNA molecule. So you have the phosphate sugar, backbone in each of the vertical portions of the DNA molecule. I have enlarged a small portion here. Wherever there is sugar, there should be a nitrogen base. Suppose here it is adenine. In the opposite chain, in the same position, it should be thymine. Adenine pairs only with thymine. Suppose it is thymine here, it should be adenine here. Between adenine and thymine, or thymine and adenine, there are 2 weak hydrogen bonds. Suppose there is cytosine here, then in the opposite chain there should be guanine. Suppose there is guanine here, in the opposite chain it should be cytosine. Between cytosine and guanine, or guanine and cytosine, there are 3 weak hydrogen bonds. So this is what we mean by highly specific base pairing as per the idea given by Chargaff. As a result of this, the 2 polynucleotide chains of DNA are not identical. The 2 polynucleotide chains of DNA are not identical, but they are complementary to each other. So this is the idea given by the Watson and Crick model, or the double helix model, to describe the structure of DNA molecule. Coming to the functions of the DNA. Functions of the DNA. In all living organisms, DNA functions as the genetic material, and as we have noted, right from bacteria to human beings, DNA functions as the genetic material. DNA synthesizes RNA. The other nucleic acid, RNA, is always synthesized from DNA. DNA provides the code. DNA provides the code necessary for the synthesis of proteins in a cell. Remember, ribosomes are described as the protein factories in the cell. In the ribosomes, amino acids are assembled to form protein molecules. The assembling of protein molecules is based on the code provided by the DNA molecule. And DNA, being capable of replication, can pass on its own copy to the succeeding generations. These are the most important functions of DNA.